All right, start from scratch. We're going to do fermentation and just go through this. So what is fermentation? I'll talk about the history, where it comes from. Talk about why yeast utilize sugar, what do they do with it, and then how do they do it? How do they get it in their cell? How do they convert it? How do they work as an organism? We'll talk about how they put sugar in their bodies. I'm going to talk to you how we use nitrogen, what its purpose is in wine, and sugar metabolism. And then I also want to talk about the ideas of respiration versus fermentation, because they're two different things. Because yeast are what's cool, they're called a facultative anaerobe, which means that they can either breathe or they can work anaerobically. They can go either way. They're kind of like the salamander of the microbe world. And then we'll talk about what we need to do in order to make sure that the yeast have a chance to finish the job, because it's really hard. So back in the day, we used to call it zymology, because that's what Pasteur invented. You gotta realize, 150 years ago, we thought that wine just had spirits in it that came alive and made things happen. We had no idea about germ theory. We had no clue yet. We didn't know, I mean, you think about it, that it's only been 100 years that we've had antibiotics. Just the number, yeah.
Oh, it's just a fun fact. Zymology was the word of the day for Wiktionary the day our boot camp started.
For real?
Yeah, zymology.
It's a side. It's an
It
omen.
is.
I like this omen. And fermentium from Latin means to boil. And wine boils without any heat. But the reality is it's just oxidation of organic compounds. And a scarlight is using an endogenous electron acceptor, usually an organic compound. Yeah, easy, piece of cake. We'll quiz you on that later. But he was the first phytozymologist in 1857. He said, I am of the opinion that alcoholic fermentation never occurs without simultaneous organization and development and multiplication of cells. If asked what consists of the chemical act whereby the sugar is decomposed, I am completely ignorant of it. So basically, he finally got a microscope, could look and go, grape juice doesn't have these things in it, and now there are these things here. But I have absolutely no idea what they're doing. They're alive and they're thriving, but I have no clue why they're doing what they do. And then Edward Buchner got the Nobel Prize in chemistry, and he was the first guy that came up with the idea that there was something called zymates. But he thought that yeast were alive and that they put something out into the juice that broke down the sugar. He didn't think that they were eating it. He thought they were just putting something out, and this thing that he put out would break it down. He thought it was called zymates, is what he called it. So what actually does happen? And I think you've heard me say this before, but I think it's going to really start to become apparent as we go through and as you smell fermentations happen. If you haven't before, for those that do, you smell grape juice, it doesn't smell like it much. And then on the other side, we get something really complicated. So our inputs, we have sugar, we have amino acids, and oxygen, or not. We can add oxygen, and there's times that we do. And oxygen can be a very, very useful tool. And in white wine, we don't use it a lot. You can once fermentation started, but generally we don't. But in red wine, we do add oxygen. We like oxygen in red wine. Because usually we're going to be fermenting the higher alcohol. Now oxygen helps the yeast, and also oxygen helps develop that aldehyde, which knits tannins together. So we're a big fan of oxygen in red wines, but let's just leave that alone for now. But on the other side, what's crazy to me is we get CO2, we get water, we get hydrogen sulfide, and hydrogen peroxide, we get energy because ferments get warm. That's why you're putting it in the fridge, because they're exothermic, they generate heat. We get esters through baby alcohols. It's basically before you get an alcohol, you get an ester. And then glycerol, which you use roundness. We get carbonyl compounds, which are all these other things in wine, like aldehyde and pyruvate. Thiols, which are the passion fruit and guava aroma in Sauvignon Blanc. Can also create sneaky aromas, like hydrogen sulfide that smells like rotten eggs. Maybe one of our ferments will be lucky enough to do that, and we'll have to correct it. If you don't correct it, it smells like the rosé. With rosé, it's coming around. But literally thousands of compounds. We go from something that has a modest number of flavor and aroma compounds to something that has literally thousands. Which is really, why do yeast ferment? How do they make energy? That's what they want to do. They want to go alive. They want to live. They want to live. And they do it through glycolysis. And then step one is so in all yeast, what they do is they take sugar and it runs through all of these enzymes, and then they like to store things as energy. Like the equivalent of fat in a yeast cell are these other carbonyl compounds. They're just carbon-based things. And their equivalent of fat and things they hold on until they need energy is pyruvate, and then eventually they make aldehyde, and then eventually they make ethanol. And each of those steps makes energy. And that's what they do to store energy in their system. They will ferment and anaerobically respire even in the presence of oxygen. We call that the Crabtree effect. And the reason why we care about that, why it's so completely effing mental, because it is absolutely against all the laws of nature from an energy standpoint. If yeast decided to breathe, they could make 38 pieces of energy, or units of energy, for each sugar they bring in. But instead, they choose to ferment and only get 2 units of energy, 2 for each one. Now, if you had a chance to go out and turn $1 into $38, would you do that over and over and over again? You could compound that into Warren Buffett status pretty quickly. But if you only get the chance to take $1 and turn it into 2, it's a lot more work. Why do you think yeast do that?
Instead is toxic ethanol, which causes microbial competition.
You are correct. Good job. So their job, basically, when we make wine, we're conducting warfare. We are literally conducting some sort of international relations where we are pushing species around to give us what we want. It doesn't change much, I guess, being a human. The more you know about microbes, the more you find out that being a human is fairly similar endeavor. But the goal is if the yeast can get started and get out ahead, and they can get to 2, 3, 4% alcohol, there's not much else that can live. It knocks out their competition. They can be the only thing that rules the entire roost. Do they want to do that? And that's their microbiological warfare weapon. They make alcohol. We make mustard and sarin gas for toxic warfare. They make alcohol. And that's really why we do it. So the deal is, how, once there's sugar out there, do yeast get it into their cells? So I want to talk about how we get sugar into a yeast cell and what they're doing. Like, this is what they're doing right this minute. This is all happening right now. And so the first one is we got to transport the sugar into a cell. So we have the membrane cell structure of a yeast cell is really interesting. It kind of looks like your fingers interlocked together. Do you see each of these molecules right here? That's cholesterol and agosterol. You ever heard of cholesterol before? Yeah. Yeah, do you know your entire brain's made out of cholesterol? Your whole brain, all cholesterol. I have questions about things that have to do with the amount of cholesterol in our body. That's for another time. But we want to build cholesterol. And the way that we build cholesterol for yeast is yeast do it by breathing. So when we're bringing yeast to life, we tend to want to add oxygen. And when we do the sparkling wine, when I start, I won't be doing it anaerobically. I will have oxygen that I'm pumping directly into the system while we're bringing it back to life. You didn't have to do that because the yeast you used came from a factory and they pumped them full of oxygen before they dried them up and put them in the freezer. So they're already adapted, ready to go. That's what dry yeast culture is. But reality is this membrane structure, because it's closed, limits most things going through. It's like your skin. Basically, these little transporters where they let the sugar in, it's kind of like your mouth. It's where they eat. And there's 3 different ways of transporting. We have one called diffusion, we have facilitated diffusion, and we have activated transport. We'll talk about all 3 of them and what the point is. So diffusion, number 1, low energy, super easy for the yeast. They're just hanging out. It's like sitting at a sushi, you know the sushi trains? As they come by, you don't even have to order, you just stand there and put food in your face. That's basically what it's like with diffusion. The downside is that it does operation down basically concentration gradient. Inside the yeast cell, there's no sugar. There's none. They're pH neutral, pH 7, and there's basically no sugar inside. So what happens is when there's a whole bunch of sugar outside, it'll just flow into the yeast cell. Call that osmotic pressure, osmotic stress. Now here's the problem with yeast cells. It's a bit of a challenge. Is that if our sugar content gets above about 24% sugar, the osmotic stress is so much that the yeast have no control over how fast that food's coming in their system. So do you ever see that like Lucille Ball segment where they're trying to eat the chocolates as they're going by and she just can't eat it fast enough because she shoved her face full? Well, that's essentially what happens to a yeast if there's too much sugar around. It just gets force fed. They become like the foie gras of the yeast world. And instead of getting a fatty liver, they don't have one. Sorry, they're pretty small. They're only single cells, no liver. Sorry, property. But what they do is they will freak out and they just make volatile acidity. They make acetic acid. Yeah.
Did you say that happens when the yeast is over 24 breaks?
Yeah, that's about 24 and a half is somewhere in that range, and everyone's a little different. But we do know that above 27 it's pretty significantly stressful for them, and above 30 is really stressful. And so what can happen in that case is you have this issue where the yeast, because they're being force-fed, they're making something that's toxic to them. Ethanol is toxic to them too, but not as much in the long run as it is to other things. So they're doing the short-term game, playing the short-term game to win, even though they know in the long run it's going to kill them. I take that humans are here to do the same thing. We can take it on two fronts. I'm going to take the George Carlin approach. George Carlin basically said that we are here as humans. We were inoculated onto this planet full of oil to make plastic. So instead of making alcohol, we're here, we're going to make plastic. We're going to eventually make so much plastic it's going to kill us. But then the aliens can come back and harvest the plastic because it's probably a really good drug for them. So just like us and alcohol, maybe we are just the thing that was inoculated on this planet to make plastic. And instead of fermenting sugar, we're fermenting oil with the plastic. And then it'll eventually kill us in the long run, which all evidence shows that's probably right. Maybe we're not as fancy as we think we are, right? I always like to take it back to that bigger picture thing. But if they make VA, the problem is once you get up around 2 grams a liter volatile acidity, it's really toxic for the yeast. They don't like to live in it. So they're even taking a more of a short-term game to make VA. So in dessert wines like ice wines, chocolate Auslese, things that are botrytized wines with really high sugar, those wines usually stick. The ferments give up, and they stay naturally sweet. And throughout history, those are the most prized wines. Dry wines are a new phenomenon. I do not want you to think that dry wines are like a thing that we were going for. It just sort of, they did go dry. We didn't really get a choice. We couldn't stop it. But so every wine was dry. But if you got a wine with sweet, natural sugar, talk about things that have been prized throughout history. It's something we haven't left of. But it was something really special. So sweet wines were always the favorite thing we had. Matter of fact, Romans would put lead in their wine because it wouldn't re-ferment, but it tasted sweet. You know how that went. Point being, diffusion is what happens in high sugar concentrations. Facilitated transport. This is also known as facilitated diffusion. It's limited energy requirements. The thing is it requires these things called permeases. Permease. It's a thing that breaks down, bring the thing into the cell. Again, it operates down to concentration gradient. We have to have more sugar outside the cell than inside the cell, which for the most part during fermentation is the case. This transport doesn't require energy. It just sort of requires these little permeases that we make. And they make these permeases out of thiamine, and they make them out of oxygen, they make all kinds of stuff, but they make these permeases, and they make them out of nitrogen, which is why we care about nitrogen in the wine. And this is the preferred way yeast like to live. This is how we like to live too. We want to go get a burger when we want to go get a burger. We don't want to worry about it. We want to have enough money in our pocket that that's not an issue. We don't want to be struggling. We want to be able to, have a meal at lunch or whenever we so desire. We just don't, we want to eat when we want to eat. And that's kind of how the yeast work. They're like, I need a little energy now. I'm going to go work a little bit. Let's go get some food, And that's how they work. This is where they want to be. Because they're controlling the flow of sugar into their cell. They're using all the enzymes around, they're using all these enzymes around, all the nitrogen around to produce energy and live, but they're living a pretty high life. This is like utopian living right here. Got everything they need all set up. And then finally, we end with active transport. This is us after a nuclear holocaust. So what happens is, eventually, the sugar depletes so much that there's not much left. There's less sugar outside the cell than inside the cell. And now, yeast have to go scrounge for food. They're going into full-on hunter-gatherer mode. Not only are they going into hunter-gatherer mode in a traditional sense, because they're going to have to look for the sugar, they're going to have to actively bring that sugar into their cell and burn more energy to bring it into their cell while they're chasing down their food, because there's not much left. They're also doing it in a high alcohol environment, which sucks. Because alcohol breaks down the cell membranes of yeast. So it's really hard. So they have to fight really hard to finish that and to ferment. Yeah.
The alcohol that they make.
Yep. The plastic that's going to kill us, it's basically the same thing in the long run. And then what's really going to be crazy is when I start showing you yeast curves and where we're at. Man, I tell you. It's so funny, I've been talking about this for a year, and now I'm like, oh, the word just keeps kind of coming true. I'm not sure about it, it's just an idea. I'm basically like, it's not necessary. But types of sugars, I think this is more important. So, yeast can ferment a whole, yeah.
Sorry, quick question. So those three forms of transport happens the same, not at the same time, right?
Correct.
So first the first one, second, okay.
Generally, I mean, yeah, generally. And here's what's what's really crazy about yeast. This is what's nuts. Yeast have this thing called quorum sensing. And so they will all be working on the line of just hanging out, sucking up some sugars, building up biomass right in the beginning. We saw this, you all saw this, called lag fags. We threw some yeast in and they're kind of just like, not doing much, hanging out, right? Well, they're building population at that point in time because they're building more yeast. They want to build enough yeast so they have a big enough population that's ideal to ferment that point. And then, in seconds, they all switch. Every yeast cell goes through a genetic shift, and boom, they all start fermenting. Message goes throughout the whole population. It's time to go through chemistry. They reach a quorum. That means, like we have in meetings, you reach quorum. You got enough there to do the job. Time to do the job.
So what happened with us is that they didn't get the message and they didn't start fermenting because we're 2 days behind?
No, you're fine. They did exactly what they did. Well, sometimes, you know how sometimes it takes longer for the people to show up to the meeting for quorum?
Oh,
Yeah. Sometimes they're all there at the beginning, and sometimes you gotta wait a half hour. Yeah, it's like, it's funny, yeah.
So we didn't have quorum?
Didn't have quorum. They were a little slower. And you used a much less aggressive strain. So, too. So, I mean, you guys were using the VIN 17, which was my stuff, you cross-pitched it. And it's not a very aggressive yeast. It's fairly slow. So it takes a little while. It's known to have a longer lag phase. Whereas QA23 is like, let's go. And then in the case of the orange wine, I think they had some stuff that had survived being in a freezer for 2 years, and it was pretty badass and ready to rip. And so they put it back in and it said, hi, let's go. I'm not frozen anymore, it's time. That's my guess. I think it'd be fun. I'll make sure I can do it, but we can do DNA profiling of yeast with ETS, and we can find out exactly what species are in each fermenter. So I'll ask them if they can do that. It'll be a retrospective thing, but we'll see if we can do that.
Would it also that native yeast was more present on the skins and stems than anything else?
I think, yeah. Well, or just stuff was hanging out on the bottom of the container because you got everything else.
You just pick the right one.
Yeah. Mm-hmm.
Or right one.
Maybe, yeah. I'm trying to remember. So we put everything, we wrapped everything to tank, and then put everything back in carboys afterwards. So they should be fairly even. But I just think that little bit of yeast and stuff that you drag out of because I rack off all that stuff. Let it settle, and then we fill the carboys. So I'm guessing that little bit of extra stuff that you have has some stuff in it. So Saccharomyces will use a whole bunch of sugars. They like primarily glucose and fructose. Glucose is their favorite, just like us. Our favorite is glucose. We like glucose. Glucose is our favorite. If you ever met somebody who's a diabetic and they're low blood sugar and you gotta hit them, you hit them with glucose. Glucose goes straight into your bloodstream, is what you use as energy. Fructose has has to be converted slightly. It has to be sort of flipped around in a direction. It takes a little more energy to use fructose. So most yeast prefer glucose just slightly. Fructose they don't like as much. You may have heard that humans may not want to eat as much high fructose corn syrup because we have to process it through our liver before we turn it into glucose. And then sometimes it stays in our liver as fat. And then that's the beginning of a really bad ride. So glucose is one. Then we have things like maltose, which is a disaccharide, sucrose, which is what we get from the grocery store. Sucrose is glucose and fructose, it's just stuck together. But the yeast have to break it down in order to use it. The good news is sucrose breaks down in acid. So if you put it in wine, it'll just start to break down into glucose and fructose anyway. A process called acid hydrolysis. Don't even know that, just so you know. Or not, right. Nice, no. And then things like maltose. And these are things that we have in beers. These are the unfermentable sugars that they can get through, but sometimes they don't. And this is why beers end up sweet. It's because they don't have just glucose and fructose. They have a much more complex pile of sugars to get through. And that's why it's a little easier to stick a beer ferment than it is to stick a wine ferment. Because they have more sugars that just can't be fermented. All right. Now we have nitrogen. Jesus. Sugar is what they eat, but they need nitrogen to eat it. So why do we need it? We need it for yeast growth, build biomass. We need to have it around. They use it for metabolic functions. They make alcohol. There's a lot of things stuck to amino acids. We have lots of amino acids in wine. And so some of the things that get chopped off of amino acids, because grapes are kind of sloppy in what they do, or maybe they're really precise, I don't know. But they dump a whole bunch of different compounds attached to stuff into wine. And so I've always used the term ammoniated, the things that are stuck to an ammonia molecule, instead of glycosylated, ammoniated. And so they make different aromas from different amino acids. Sulfur metabolism, so do they get stinky or not? And this is something they really worry about in beer. We don't worry about so much in wine. And then we also can make ethyl carbamate, which I'm not going to talk about anymore. I'm going to say it for one thing, and one reason only. I'll bring it up and I'll cover this for a moment. So one of the things that got brought up, see what happens sometimes in science. In science, sometimes people get really expensive new toys, and then they find out they can measure a thing. And then when they measure that thing, they get really excited about it and publish results about it. And then it turns out the thing that they published was really pointless. There was a big hubbub about, I don't know, maybe 10 years ago. I think it was about the levels of arsenic in wine. And that, arsenic? I think it was arsenic. In wine. And there was a lab that got a new machine, and they're like, there's arsenic in wine. And I can't remember the numbers, but they're like, and the big producers are the worst. They have more arsenic than anybody else because they grind the seeds up a little more and there's some arsenic in the seeds. And then they start talking about it, and they're just, literally this blog gets blown up, and it's going, it's hitting all the major press that there's arsenic in wine, and only major producers are bad, and 2 buck chuck is terrible. It turns out the rates, I think, were like 50 times lower than the strictest drinking water standards on the planet. So it's just because you can measure it doesn't mean a damn thing. So yeah, they did, but it was not an amount that matters. And as a matter of fact, you need a little arsenic to live anyway.
I think they were saying the same thing about chocolate for a little while.
Yeah,
Eggs.
tis not the tis not the compound, but the dose. Guess what?
I mean, Cheetos has red foliar, and everybody eats it. I mean,
Nope, nobody, you know what? Nobody gets out alive anyway. Give it your best run, have some fun while you're here, right? It's always been bad for me. So ethyl carbamate, turns out if you add excess diammonium phosphate to your wine, it sticks around, it hooks up with some of the dead yeast and alcohol, it mixes a compound called ethyl carbamate. Ethyl carbamate is a listed known carcinogen in the state of California because pretty much everything is. But, so they published this article and everybody's talking about ethyl carbamate, you can't use this, none of ethyl carbamate. Well, Gordon Burns did the study, and it turns out that if you make a piece of toast, you end up with about 1,000 times more ethyl carbamate, because when you toast the amino acids on a piece of bread, it's like infinitely more than would ever end up in wine. And God forbid if you grill meat. You sear a meat on a hot stove and those amino acids get hooked up with some heat, they just get turned into all kinds of ethyl carbamate. So it turns out just because you can measure it doesn't necessarily mean it's a big deal. It was the hubbub for about 10 years, and we found out it was pretty much pointless, and, well, ended the ethyl carbonate thing. All right, nitrogen. We're going to talk about two things. So we talked about YAN. You've seen me talk about it in class, and we can talk to you about what it means a little bit more. YAN means yeast assimilable to nitrogen. That means it's nitrogen that yeast can use. There are 20 amino acids. Yeast can use all 20, but do not. They leave a couple behind. And yeast have a preference for different amino acids. Amino acids, if you think about it, are all the things that we need to produce proteins to live. We have high levels of amino acids and things like meat and chicken and stuff like that, poultry. Also beans have high levels of amino acids. But they're the things that we need to be able to kind of survive and say we need. So here's the preferences. I forgot my button. That's not a pointer. It's not a pointer. Not a pointer. Got to break my own habits. So the thing is I'm going to show you that top line. There's one called ammonium on that line. Ammonium is not an amino acid. Ammonium is free nitrogen. It's very available, and yeast love it. It's their favorite nutrient. It's very accessible. They put it in their shell really quickly, and they like it. So here's the deal, is that hours to 50% depletion, basically in 16 hours, all the ammonium in a fermenter is gone. It's actually faster than that if it's a live fermenter. My dad and I thought we had figured this all out because we add ammonium to our wines. So we put an ammonium probe in our fermenters, and we were going to just keep the ammonium levels at a certain level in order to keep the yeast happy. So we'd add some, and in like 20 minutes it'd be gone. So what's crazy is that yeast are basically feast or famine. So even though they like to control the amount of sugar that comes into their cell, in order to be able to control what comes into their cell, they want all those amino acids. Because those amino acids are what make all those permeases that allow them to eat at their own rate and everything else. So they need to bring those in. So whenever they go find this stuff, they go scrounge and get it yesterday. So they want it quick. And so this is a particular conundrum in fermentation because the first generation that really takes off takes all the amino acids and puts it in their cell. And then the next generations that come along don't eat it. And they don't leave anything behind until they die and spill their guts, and then eventually they put it back in wine. Because your YAN won't go back up after they're dead. So I guess maybe there's some cannibalization going on. But again, there's some references to humanity here. I don't think there's like an instance though in fermentations where one yeast takes 90% of the amino acids and leaves 10% for the everybody else's. Maybe there is, I don't know. Maybe there's a collective of yeast that steal it all, I'm not sure. Anyway, I'll leave that bit of philosophy aside for now. It takes 1 mole of ATP to transport nutrient in the cell. If ammonium is too high, yeast shut down. So what's interesting is yeast, while you're fermenting, if you add a whole bunch of nutrient up front that's pure ammonium, which is diammonium phosphate, that's what we usually use in winemaking, it's because it's super cheap. If you use it early on and there's too much, the yeast will never eat the amino acids. It'll turn off, they'll switch off and be like, this is too easy. Like, this is just delicious. I'm never gonna, stop just shoving, this in my face. I like this a lot. So if you do that, it'll shut down the use of all the other amino acids, and they get left behind in the wine. And so you want them to get all consumed. We want those to get eaten up. But we can see the things that they like a whole lot, and then we have some things that they don't like glycine, because they just don't like it. I'm not a big fan of tyrosine, and this one's important to remember. And then we also have this guy, and this one's really important to remember. So notice the final utilization in the wine of this one, histidine, and tyrosine leaves a little bit behind. But the reason why that's kind of important to note is these are, if you chop a carbon off of histidine, you get histamine. And if you chop a carbon off of tyrosine, you get tyramine. And those amino acids are converted by bacteria in secondary ferments. Things go through mal or some other bacterial fermentation. And those can be the creatures that leave us with things that are very toxic to us, things that really probably are the major cause of red wine headache and things like that. is these high amine wines. And you can, fortunately, get around it if you take a Benadryl before you have a little bit of wine, but then you might just pass out on the floor after one glass of wine. So, you know, just one of those things, but something we like to talk about. So, what we always do is they really like ammonium with lysine. And then, again, each one yields between 1 and 4 units of nitrogen per amino acid. Arginine only contains 4, whereas tyrosine only contains 1. So it's like, to eat tyrosine, it's a direct expenditure. You know, in order to eat that like little tiny salad, it's like 150 calories, you had to go run 2 miles, you know, in order to get it. So most people don't like it a lot. The ammonium is too high, yeast to shut down. So anyway, again, this regulation of yeast. They provide like sources of great growth preference and order. They're great for things ammonium, and then amino acids, and then they'll need amino acids residues. But again, I put that squirrel with the nuts in its mouth because they are feast or famine. They want it, they want to fill up their face, and hold on to it. Surplus amino acid is stored in their body, like fat. It's just stored, and then they use it when they need it. So how does nitrogen fit into this whole transport thing? Well, nitrogen amino acids are required for protein synthesis. Enzymes are proteins. So they need protein to make the proteins that they need to bring the sugar in their cell. That's pretty much it. We make big fancy words for it, but they need protein to make protein that they like to use to make the sugar come into their body. That's pretty much it. And so they require nitrogen. Well, the problem is nitrogen runs out, and yeast can't eat. And when they can't eat, they struggle. The new thing is they don't die. Yeast are crazy. They'll start making amino acids out of shit going on. They'll start clawing around and finding things, usually based on sulfur. They'll find it and start chewing it up in order to make something to keep going. And then in the process, they make rotten egg smells that smell like hydrogen sulfide. So if you ever smell a wine that's getting real stinky, you know, there's a reasonable chance that the yeast are starved. And they're giving you a notice that, hey, come help me, give me some nitrogen. And we can do that. We can supplement with nitrogen. We have all kinds of yeast nutrients we can use. You can buy many off the shelf in a whole bunch of different ranges and styles. And everybody will tell you theirs is the best, but they're all the same damn thing. Pretty much. They're all basically dead yeast, because yeast are cannibals and they're sick like that. Or they have a little bit of yeast and diammonium phosphate, and maybe some other vitamins just to make you feel good. But they're all about the same thing. But of course, they're specially formulated and unique and whatever.
Basically, that's what sterile flash is, right?
Sterile flash is probably dead yeast. I don't know exactly what it is, but I think it's like highly refined sterile. It is actually a little different. I feel like that's one of the few things I feel like it's been worth my time to invest in. I feel like all the other ones are kind of bogus. They don't make dead yeast. But that stuff, they did do something special with that one.
Sterile flash is the real deal?
It seems to be. It's made my life a whole lot easier. And man, those yeast do sure bloom a little better. And I used it the other day in some bread. And man, it got so fluffy and light. And it was like I used built like a really serious culture. So I don't know. The sterile flash, the stuff that we added before when we made our yeast, you didn't make yeast oil. But we made, you'll see it when we do the sparkling wine. But we make a little add to the water beforehand. We stir it up, and it's a really high sterile, which means it has all the cholesterol and agosterol in it that the yeast need to make their cell membranes. And then you throw the yeast in, it starts, and basically you just go pitch it. What you used to have, so let me give you an order of my life in the wine world. In order of my life in the wine world, if you wanted a special yeast, you had to buy it on a slide. And then you had to build up a culture. Took you 3 or 4 days to build a nice culture, and then you could pitch it. Then along came active dry wine yeast. This is in my life, and we got a couple of different kinds. We had like Premier Cuvée and like EC Triple 9. It was like 5 kinds. And we were like, oh my god, because you could take this, like the stuff you use for making bread, you could just pour it on top, and it would bloom, and then you go pitch it in the wine. But the thing is you had to, when you did that, you'd pour it on top of some water, let it slowly settle in, wait 20 minutes. Then you'd have to temper it for 20 minutes, another 20 minutes. You'd have to do this for about an hour to 2 hours to get the yeast culture all set up, ready to go in the kitchen. So it's taken a 2-hour process down to 20 minutes. So it's gotten better and better. So that stuff's pretty neat. But sterols, all that stuff. So all the outside of these is all membrane. All the stuff that they need to adapt and be tough. That cell membrane is like, it's your skin, man. Without it, you don't get last long, neither do they. They need a really robust skin. So, consequences for flavor development. God, I don't know if I do this or not.
Glutathione is anti-aging. That's like reverse aging.
Yeah, supposedly, yeah, but yeast like it a lot. It's a very powerful antioxidant, but it goes away really fast in wine. And we try to protect it a lot when we're doing white wines. It's a useful indicator of oxidation because it goes away if it oxidizes really quickly. We try to hold on to it, not necessarily for yeast health so much. I mean, it does help them a bit, but primarily because it's an indicator of how much we've kept cysteine around. Cysteine will oxidize, and some of the compounds on cysteine will oxidize, but more additionally, the cysteine isn't an amino acid that yeast like to eat a whole lot. It's sulfur-based. They don't enjoy it. It's like, I don't know, kale that we've all convinced ourselves we like, but we really don't. I grow a lot of kale and eat a lot of kale, but it's really, I mean, I've had to convince myself I like kale. Yeah, but it took time to convince yourself. And so the thing is in a fermentation where we're trying to drive that style of peach, or the guava, passion fruit smells that you get in Sauvignon Blanc, we have to force the amino acids to go there. We have to have the yeast to go to get those amino acids. We have to force it. So what we have to do, this is really hard when you're doing like Sauvignon Blanc or you're doing Pinot Gris. You gotta let the ferment happen, and then you gotta let it get stinky. Am I too loud?
I was just peeking in, sorry.
Oh, no, I was just making sure I wasn't blowing you out.
Oh, no.
So what happens is you let the fermentation go, and then what happens is they finally gone through all their amino acids, and then they get to the methionine and cysteine, and they're like, oh fuck, I like let this game. That's all I'm gonna do. God, but if we want to keep going, let's keep going. So they eat those. The interesting thing is the grapevine in Sauvignon Blanc grabs these aroma compounds and sticks them to a cysteine molecule. So what happens is the yeast is like, I don't need that aroma molecule, but I want the cysteine, so I'll use it and eat it, and they cleave that aroma off. But the thing is while they're converting sulfur-based amino acids, the wine starts to get a bit stinky. So the challenge is you got to ride this like razor edge of letting the wine get reduced in order to release all those aromas, but you don't let it go too far so that it gets so stinky you can't recover the wine. So it's like this real razor's edge. And so what usually I would do is, when I do Sauvignon Blanc, because I'm watching for it smelling, and I usually know somewhere where that's going to show up. In Washington, we don't have a lot, so usually around 15 bricks it comes off. But in New Zealand, it was killing me because everywhere I walked, everything smelled like stink because they were letting everything get stinky. And then around 10 bricks, they bring a bunch of nutrient and they would blow all the hydrogen sulfide out, the stinky stuff out, so it would leave all the cool passion fruit guava aromas around. So you have to sometimes restrict the nutrients to make them eat their veggies so that they'll make the cool smells. And if you eat enough kale, you'll make smells too. So anyway, that's what I just said. But you just gotta be careful. But if you use too much DAP and you just go ahead and start feeding these things all the time, you never force them to go down and eat those amino acids, you won't get those aromas. The only two major varieties I know that do this are Sauvignon Blanc and Pinot Gris. That's kind of it. Sauvignon Blanc's the big one. Cabernet Sauvignon actually has a lot of thiols too. Cabernet Sauvignon has a ton, huge amounts. If you ever make rosé with Cabernet Sauvignon, you make it like Sauvignon Blanc, it'll smell like Sauvignon Blanc. The reason why you don't smell it in red wine is because sulfur compounds stick to tannins, and you can't smell them because they're stuck to your can. So they get stuck in the matrix. They can't get out. So yeah, you liberate them and then they get stuck. So anyway, that's why you never had a passion fruity guava cap sauv. However, if you ever do make rosé out of an overcropped Cabernet Sauvignon vineyard, you can make some very Sauvignon Blanc Cabernet Sauvignon rosé. taken 5. I keep you up for almost an hour. So, go get a drink and talk amongst yourselves and wander around. Huh? The phone? Oh, yeah, cell phone. Thank you. And again, all right, now that we're done with the conspiracy stuff, let's do sugar metabolism. All right. So I'm going to do a little thing. All cells require energy. So I'm going to do this real fast because you don't need to remember this. You probably haven't had biochem. Anybody here have biochem? Well, yeah, sort of. Okay, good. Yeah, that's it. Oh, yeah, you're in. There you go. So I don't need to go that far. So basically, what we have to do is we have to make sure that we have ATP, adenosine triphosphate. And this is what we make. And like the reason you know ATP is, say you go out and you, it's actually it's a good comparison for 2 sports that I won't play both of. So one involves using up your ATP, and the other one involves recycling your ATP, even though they're very similar sports, and one's football and one's rugby. And so rugby, you're moving all the time, and because you're burning it all the time, you lose your ability for maximum impact. You can't hit that hard because you're just sort of worn down because you don't develop all your energy. You only need about 45 seconds to regenerate all your ATP. So in football, we get about 45 seconds between plays to regenerate everything and then go out and smash somebody's head off. And that's why the collisions are just so much more violent in football. Also, I think pads actually make it worse because you aren't going to check yourself if you got a helmet on or you got a shoulder pad. You're going to hold back on wailing somebody. But when you're in rugby and you're leading with your head, you're like, do I really want to hit that guy's knee with my skull? Probably not. I'm gonna jump a little bit. But anyway, ATP is a big driver in that. So, but that's what we all thrive on. It's what we use energy. It's just how we move through it. But basically, when oxygen is present, this is how things happen. So what we do is we have glucose that comes in, and 2 ATP go here, and then we burn a little more ATP, we make a little more ATP, and then we end up with pyruvate. So the net gain, so this is glycolysis, this is during sugar, so we're making pyruvate. At the net day, we only get 2 extra units of energy. So let's do cellular respiration. This is a little different. So if they can breathe, we bring in glucose, they make pyruvate, and then they make all these other things. They make the citric acid cycle. Remember I talked about that? It's also known as the Krebs cycle. And then eventually, they just leave a little bit of, well, all that would happen is they would turn that sugar into little oxygen and water. And that's it. But they make 38 ATP in the process. They would make so much more. But then, yeah, life would be pretty boring. Where's the booze? So you don't get it. So the good news is for us, is yeast decide to sacrifice themselves at the altar of alcohol, or else our jobs would be very, yeah. So anyway, back to my coffee here. There we go. Okay. Very good. So I'm just going to do fermentation. We did that. So let's go ahead and do this. Oh, sorry. Anyway, I have an extra slide there somehow. It just repeated itself. During fermentation, 95% of sugar is converted to ethanol. That's amazing. 95% is converted to or CO2. Ethanol CO2. Then 1% is converted into cell biomass, then 4% is converted into other stuff. Pyruvate, aldehyde, other stuff. And then one thing that's also nice to know is that you get a 3-degree temperature increase, all things being equal and closed, per 27 grams of sugar fermented. Now 27 grams is 2.7 grams. Because it's in thousands. There's 1,000 grams in a liter, so 27 grams in a liter. So if you had a 27-degree brick wine, you'd have about a 30-degree temperature rise from whatever it started at. Yeah.
So your wine will, for every 27 sugars, it will raise 3.
4 up. Do you remember how high your red ferments got? Yeah, they're cooking. That was all natural. They're making everything. But yeast are like you and me. Right? I'll say this again every time. We're all born out of the same soup. We all come out of the same stuff. We all like the same temperatures. So yeast ideally will function as yeast aren't like us. Like just got a coat on. Yeast can't smoke. Get a little cold. They they live in the same temperature we would live in if we didn't have the ability to warm ourselves up. So realistically, depending on the person, somewhere between 55 and 95 degrees is about where we're at. Depending on the person. Some people like it a little warmer, a little colder, but most things like it around 70. Yeast really like to ferment at 70 degrees. They kind of cruise right along. It's kind of their happy spot. Don't move. And the way that we slow them down is we slow down all their chemical process in their system. We slow down temperature. We can slow them down by putting them in cold rooms. That's why we chill tanks, cool things down, slow the process down. If we want it to go faster, we can warm it up and let it go a little quicker. Or in the case of red wine, in 1.5 ton ferment, you're basically going to go up about 20 degrees from baseline, 20, 25 degrees. And if you're starting at 65, 70 degrees, and you go up 20 to 25 degrees, you go up to that 85 to 90 degree spot, which is where we really like to be. Because when you make red wine, I'm going to give this to you guys because you haven't heard it before, but basically, the phenolics that are in red grapes, in shape and structure, are virtually identical to that in tea. So if you make tea, what kind of water do you use? Warm water, yeah. And the warmer it is, the better those tea bags extract, because that's what we're doing is dumping them back in wine over and over again. Then the other thing that happens too is that also is really good at extracting. If you ever put a tea bag in vodka, you'd be amazed at how quickly all this stuff comes out, because alcohol is also a very good solvent. It'll pull all those things out. So we get heat and we get alcohol together, we can really pull a lot of color, flavor, tan, and all that out of the wine. So that's how we kind of manage our red wine ferments and how we drive extract. And we don't have to use a lot of exogenous heat if we just can use Mother Nature to our advantage. And I'm a big fan of that. When you can use less energy to it. This is the picture. This is what actually happens. So this is glycolysis. And anyway, I'll just do this really close and simple, but this is all the stuff that happens in order to make energy. So if we take glucose, it comes down this pathway, and then we go all the way down here to pyruvate. And all I care about that you ever remember is glucose to pyruvate. None of the other stuff I'm never going to think about. I've never stood over a fermenter thinking about any of these things. But notice that if you do any other sugars, there's extra steps involved. And every extra step requires extra ATP and ADP generation in order to make things happen. So it just takes more energy. The more complicated the sugar, the more energy it takes to ferment. But all that matters at the end of the day, we get here. And in my mind, pyruvate to yeast is fat to us. It's an energy store that they can use for later. And they want to make as much as they can. So, this is actual fermentation, but this is where the one little thing changes. So we have pyruvate, and then we have one more step where we release the CO2 from pyruvate. This is where the CO2 comes off.
Ethanol, right?
No, aldehyde.
Oh.
And then the final step is aldehyde to ethanol. But we only get 2 ATP for each. So anyway, all this stuff right here, and then pyruvate, which is also pyruvate acid. And this is the fat, and then when they decide they want to finally cleave off and make their last bit of energy, aldehyde, that'll get turned into ethanol. In that process, CO2 comes out. So really it's sugar to here, and then this is the last end of the step. So in a short and simple world, this is it. But why this matters is these two enzymes right here. Pyruvate decarboxylase and ethanol dehydrogenase. So, remember they're ACEs? So, it takes a carbon off. That's what that means. It pulls a carbon off of pyruvate. The carbon chops off and becomes CO2. So it's this enzyme. And you know what enzyme they need to do this? What vitamin they like to feed? Thiamine. Same with this one. This is thiamine. So acetaldehyde dehydrogenase, alcohol. So this actually this is from my professor. He's wrong. This was my Adelaide professor, Paul Grubin. You're in deep shit. You're wrong. This should be acetaldehyde dehydrogenase. That's wrong. I've been giving this lecture for years and I just caught it. So anyway, it chops the hydrogen off, and then we get alcohol. The thing is this particular beast right here, in both acetaldehyde hydrogenase and ethanol hydrogenase, you make out thiamine. So the vitamin thiamine. So thiamine is what allows you to produce those proteins, is what allows you to produce it out of the nitrogen. And almost everybody who comes into the hospital with a liver that's on the last edge, they're more malnutrition than they are from the direct damage of alcohol. And usually they're thiamine deficient. And so in order to make sure that yeast finish their job, we add thiamine. Thiamine does exist in a reasonable level of grape juice anyway. You can supplement it, I recommend it. Most of the superfoods and things that you buy that are commercially available have extra thiamine added to them. But if you're in the alcohol industry and you're regularly drinking, you should probably be supplementing every day with some sort of B vitamin that has a high level of thiamine if you're going to be drinking regularly. It's a smart move from
I mean, that's B6.
B1. Yeah.
So a monster a day?
Well, yeah, I don't know if that's gonna help offset the sugar in it, but
They have sugar-free ones.
so yeah, sugar free, but yeah. So several hundred other things are done. We talked about this a little bit, but one thing I do want to say is that we can drive, and this is a suspicion I have. I haven't tested this yet. This is a swag. It's a suspicion, but I have a suspicion that when we're in higher alcohol ferments and yeast can't regulate themselves very well, in wine we don't just have alcohol, just ethanol. We have many alcohols. There's about 30 different alcohols in wine. There's methanol, which comes from the grape skins of all things. Then we have higher alcohols like propanol, butanol, butyl, and these alcohols are, we call them fusel oils. They're big alcohols with more than 2 carbons. And I have a suspicion that when we have high alcohol or high sugar ferments, they make more of these other alcohols. And our body has a hard time processing them, which is why some people have a hard time, me personally, with higher alcohol wines. I can drink booze, fine, because it's, vodka is fine because it's pure ethanol. But if it has to be purified, higher alcohol wines really struggle with it because I think I have a hard time breaking down some of those other alcohols. So that's a study I need to go do. Someday I'll do it. But there's all these other things that come along with fermentation. And this is that idea of the Crabtree effect, when yeast make alcohol in the presence of oxygen. In high sugar environments, respiration gets pushed back. And this is when the yeast know that there's enough sugar around that they want to take it, they can make enough alcohol to put everybody else out of the room, so they go ahead and shut down and make alcohol instead of having a very healthy, happy, simple life. It's a choice that they make. But what's crazy is, as we get alcohol contents up, it gets harder and harder for yeast to live. So, yeast are going to make their own demise. But the last thing you ever want to do is deal with the stuck fermentation. Unless you're trying to make ice wine or something that actually does stick. Because stuck fermentation sucks. Especially if you're making red wine, you gotta take through malolactic fermentation. You get all sorts of problems with stuck fermentations. They are the bane of my existence. And so what are those things? So I'm gonna show you the growth phase of alcohol. And so this is kind of what we do in yeast. Yeast come along, and the viable yeast cell goes up, and then it levels off, and then the sugar count drops. So we have the inoculation lag phase, exponential growth phase. Right here is when they start making alcohol. Right as they plateau. Almost every microorganism that does what it's going to do decides to do it once it reaches carrying capacity on the land. Whatever it's going to do in its population is when it levels out. So whatever effect it's going to have. So then it makes alcohol, keeps making alcohol, but then the alcohol concentration gets so high that it really starts to beat up the yeast and they die. So if you want to think about what it's like to be a human, you are here. You're about to find out what we're here to do. So maybe we're going through some sort of quorum sensing right now between our collective selves trying to figure out why our world's in such chaos.
We're gonna make plastic, right?
Well, maybe, just maybe. This is our quorum sensing that allows us all to talk to each other and tell us what we're going to be doing next. I don't know. I just push it out my ass. Just yeah, no problem. Okay, so I'm going to show you something really cool. And I think this also illustrates something human. So we did, one year we did an uninoculated carbon year, where we went out in the vineyard and made this very special aseptic, what's called a pied de couve, which is basically means bottom of the bucket. But we took berries, went out with gloves, ethanol. We bought brand new buckets that had never been in a winery, sanitized them with alcohol that had never been in a winery. We lit the buckets on fire to make sure that they burned any microorganisms out like aseptic, and then went out and picked very carefully all the berries. And then we left them out in the vineyard to start and build a starter culture in the vineyard. And so what your idea is behind the pied de couve is whatever that microbiological warfare that you want to happen, you want it to happen in the bucket. Yeast will eventually win, but God knows what's going to happen in the interim. You might end up with a bunch of VA, you might end up with a bunch of other stuff. So people like to do natural fermentations. I generally recommend that they go build a culture and let it get going out in the vineyard. And then once it's healthy and happy and you've got yeast as the dominant thing, bring that in and pitch it in your fermentation. I do have an SOP for pied de couve. If somebody reminds me, I'll put it in there. It may actually be in the SOP, it's fine. I don't know. If not, I'll make sure they're done. But, so I thought this was really cool, because it's actually more complicated than you can see on this graph, because some of them are too small. But in the beginning, we had about 100 different strains of yeast. And then, as we reach that sort of peak fermentation, we're down to 3 strains. And then as the decline and death phase happens, we get some cockroaches that come out to finish it. So I think this is also a pretty cool allegory to humanity, that when we started out, we had all these thousands of different unique cultures, and then eventually we interbred, and then a few more outcompeted the others, and became a few dominant cultures. And then eventually we're going to kill ourselves off by making alcohol, and then God knows what the creatures that are going to come out in The Walking Dead at the end to finish it all. So they're the zombies that came in to finish it off, and they were all unique and different, and not one of them was commercial. They were all their own, they were unidentified. They did not match anything in the database. They were all their own unique Saccharomyces. So, because all yeast get put in, ETS puts all yeast in the database that are out there. Everything, as soon as it's done, they put it in the database and keep all the alleles, and these were all natural. I thought it was kind of cool. Anyway, so neat size sort of. Yeah.
The dark green one, it wasn't part of the first 100.
These are all different. There's more. They're just not big enough to show on the graph, but there's like 30 or 40 more that show up at the end that weren't there at the beginning or ever there. They don't share any genetics with the other ones. Yeah, you're good.
Do they mutate or do they just grow up?
There is not. Sorry. You do a relatively niche spiel. Anyway, yeah, you're right. That's exactly right. It's coming out at the end. And some other life I'll be a voiceover actor. That was what I was voted most likely to do in junior high. Anyway, fermentation basic facts. So basically, we have exponential phase, that growth phase. You've all seen that. We've flipped over. You've gone to quorum sensing. Maybe not one. Maybe one of them isn't making alcohol yet. There might be one. Might be the one that crossed pitch within 17. I think you may not be quite there yet. Maybe he's just turning over the edge right now. But we have exponential phase, we have a stationary phase, and during that stationary phase, we've got equal amounts of yeast growth to death. It balances out. And then we have decline and death phase. Then when sugar is mostly depleted. So why do we care about survival factors? We gotta get ferment done. So we have to make sure that we maintain viability of our cells. We do everything we can to increase alcohol tolerance, and we want to maintain as much energy generation as we can. So here's our survival factors: oxygen, fatty acids, and sterols. And we use oxygen to make the fatty acids and sterols. And we can do that, and when we do we restart the sparkling wine, we're going to do all this because we are going to introduce, let me back this up, we're going to introduce our yeast to their new utopia here. So we are here in the land of abundance right now. We've been living in a pretty spectacular place as humans, but what happens when our resources start to dwindle or really start to struggle? You take any of these guys here that have been living the high life and probably throw them in here at the end and they're all gonna cry. They won't know how to live, but the tough ones will come out. But we wanna make sure that we give them all the opportunity to be able to finish off. So these are the things we can give them. We can give them oxygen, we can add this, and we can add this. If we want to, this is what the sterile flash is for, so we add it to give them adequate amounts to finish their fermentation. So, anyway, I'll keep it simple. They use all those things to build what we call a phospholipid biolayer, which is their membrane. And we want to make it as tough as possible. We want to make it tough and robust. And this is kind of what it would look like in a generator plant. They need oxygen to build it up. And if no oxygen's available and you want to do something really reductive, you've got to add those sterols and things in a supplementary manner. So how does alcohol inhibit yeast? Well, there's two ways that compounds basically exist. They're either hydrophobic or lipophilic. I think no, hydrophobic or, yeah. And so what happens is basically, think of it this way, because I've just been doing this. You get a smear of butter on your counter and you try to wash it off with alcohol, it'll wash off. But if you try to wash it off with water, you just smear butter everywhere. It's because butter and water don't go together. So the phospholipid biolayer is something that can't be broken down by water. That's why yeast really like it, but ethanol really does go in and start breaking it down. Literally, and if you were to soak in a tub full of vodka, you won't last very long. I don't know how long you last, I don't want to know the answer to that. But I'm just saying it probably wouldn't be a lot of fun to break you down. So again, alcohol denatures proteins. It really? If you like crawl into that, well, you'd sing. Well, yeah, because you'd sing. It would be fun. Yeah, it would be very good for you. So anyway, basically, as soon as the yeast start to leak, we have problems. Because yeast cells, and I use all the big terms, but yeast cells are pH neutral on the inside, but they're in a pH 3 something environment on the outside. Your blood is pH 7. It drops by a quarter of a point, you'll die. It goes up by a quarter of a point, you die. If the yeast inside of the yeast cell membrane goes up or goes down to where wine is, it dies. It can't live. So it has to fight really hard. So basically what happens is in a low alcohol environment, you can see this is basically a representation of a really strong cell membrane. All it's doing is eating, making energy, having a good time. But then as soon as we get this broken down cell membrane, we have all of this acid coming in. And then the yeast goes, oh, I gotta use energy to throw acid. So now, not only have they burned energy just to live and eat, now they're having to expend energy that they've saved just to pump acid back out of their cell to stay neutral. Oh, they're just dying, they're struggling. And so, you know, what are things we can do to fix this? We can use a dry wine yeast culture. We can add the sterile flash, we can add things. We can bring back our yeast in an oxygenated environment, that will help. And then of course, we want to make sure we've got all the ample things up front so that when they bring their first population out, they've got everything they need to have a successful start. And then if you're doing reductive ferments, where we're doing, remember I was talking about just pressing with no oxygen, no anything around, because you're trying to keep the yellow, you know, the wine fluorescent green, you're going to have to be much more aggressive with all of these survival factors on the front end. The good news is, you generally don't make 15% alcohol wines that are reductive. You make 12% alcohol wines. And for fermenting to 12% alcohol for yeast, pretty easy. 15's, pretty hard. 16's, really hard. It's a really difficult environment. So even though that percentage sounds small, it's a big jump in terms of how much it stresses the yeast out. So we want to make sure we supplement. We've already done that. So, yeah, we'll go through this, do this other different stuff. More or less, I'm going to stick in white wine. So, when you do an oxidative style of white wine making, you have quite a bit of oxygen up front, because it's just getting dissolved in the liquid. That oxygen the yeast are going to use. They're going to use it to make sterols. They're going to make fatty acids. They're going to use it all up and consume it pretty quickly before they start that fermentation. But if you put yeast in a reductive environment, there's a few problems. The first problem you have is you've scavenged all the oxygen. If not with sulfur, maybe ascorbic acid, there's no free oxygen available. Those yeast are struggling. So you got to make sure that you give them what they need to get started. It's really essentially at the end of the day. So it's pretty hard to make oxygen-free wine, but we want to do our best. It does make it a little more stressful. Now, something that is interesting that I have noted over the years, oxidated ferments, really like Chardonnays that are barrel fermented, convert more like a red wine because the yeast are happier and healthier. Reductive ferments tend to convert higher alcohol. They're a little bit more like the 0.6 conversion ratio. The reason for that is that yeast, when they're under stress and duress, they'll make more alcohol to protect their environment. But when they're having an easier, more comfortable time, they convert low. It's an easier place for them to grow. So generally when you have low conversion ratios, that means your yeast will really happy. When you have high conversion ratios, you're missing something. It's just a good rule of thumb. So if you're starting to have these conversion ratios of 0.62, 0.63, like you might want to start figuring out why your yeast are stressed. But if you're converting at 0.55, and then also there are some yeast that just do that. We're starting to get better in engineering yeast because people want lower alcohol content. So we're starting to figure out some yeast that are actually less efficient at producing alcohol. So we can pick riper, but then still have lower alcohol content. Talk about that. You need them. That makes everything happen. So you can supplement with dead yeast. Almost everything's a dead. Almost everything at the wine store is dead yeast. All the nutrients, they all have dead yeast. Vitamins. These are the two that I like to do. I typically supplement. I'm not going to do anything here. I'm just not. We don't have high enough alcohol. I'm not worried about it. But I will use thiamine, and that's the coenzyme for producing acetaldehyde dehydrogenase. And that really reduces your chance of stuck fermentation. And here's my other reason for not having any problem with supplementing for that. DTB does have regulations on it, so make sure you don't go over the amount. But worst case scenario, you leave some extra in the wine, you're helping the person that's going to end up drinking it in the long run. And then pantothenic acid is basically what we do is it allows the step before the metabolic step before making acetaldehyde. It helps regulate hydrogen sulfide, so it helps keep your wines from getting stinky. So I add B5 as well. And again, something that you can use just fine. It's harmless to humans, so it keeps our hydrogen sulfide production.
Will we need to add B5 in ours, or?
No, we're not doing anything.
Oh.
This is weird. So this must be some weird. I don't know what this is. I've got multiple. I told you my hard drive would help. So you've got some extra stuff. I don't see there's anything worth me going over here. Because basically it's the same thing. It's just repeated. Yeah, I don't know what I told you my hard drive needed. But anyway. Yeah, same thing, it's just different pictures. So anyway, you gotta work. Okay. There's nothing new. So anyway, I apologize for having a slightly messed up. That's kind of like that. Questions, thoughts? Otherwise, that's a lot. Fire hose to drink from. I mean, even though it's the second time, you probably still heard a few things. What?
I got bored.
Did you?
I had a day.
Good. Yeah, you're, what the hell? Oh, okay. So, I mean, like I said, at the end of the day, we're all just organisms, and it's just, I think for me, it's fascinating. The more you know about yeast, the less cool you feel about being a person. I tell you. You know, we all do the same stuff. We all live in the same soup. Maybe I should take some more mushrooms and see how things go. I didn't have, so today's kind of a short day, I hope that's okay.
Yeah.
Was taps left out in there, or was that a different one?
Oh, we can do that. Yeah, you wanna talk about taps?
Already.
Well, that's for malolactic fermentation.
Is that
I
what
wasn't
it's
gonna do
called?
I wasn't gonna do mallow because we're doing white wine and we don't do a lot. I mean, we can do mallow. I'll tell you what.
I had it written in
Why
here.
don't, why don't you write down a little, like, if we get, like, let's say we start flowing out of, like start, like, if we do, I think I got plenty, but if we have a little short term, I mean, if you wanna take 5, I'll give you the 15-minute rundown on malolactic fermentation again. I don't know, I'd probably eat your brains 2 hours listening to these a lot, but I can do mallow. I can pull that from the cuff though. And I'll just draw a little picture on it, I'll just tell you what it does on the board, so. Actually, I will try to kind of do some, so malolactic fermentation.
Do you wanna stop your recording?
No, I'm just gonna go do mallow. It's okay. Well, yeah, as long as you won't get it, I won't put it up on the screen. It won't be in your file for later. No, let's do it. Malolactic fermentation, there's about 3 ways we can approach it. Okay? So what it is it's a bacteria, and we have lots of lactic acid bacteria around. The key ones are lactic acid bacteria that are known as lactobacillus. You know these very well because you eat yogurt. Okay? And lactobacillus usually convert lactose into lactic acid. So they take the lactose in milk and they're what make your Greek yogurt kind of sour. They turn it into that acid, and that's what makes yogurt a lot more stable on the shelf than milk, because it's been fermented. Those are lactic acid. We don't like those in wine because they make your wine taste like yogurt. Then we have some other ones like Pediococcus ganosus. It's another cockroach on the bottom of the wine world. We don't like that one a whole lot because it happens to make a lot of biogenic amines, those things that are toxic to us. It likes to make those. Also can make your wine smell like beef jerky and fermented meat, which maybe to some people they're into it, and for those that aren't, good. But the one we use is called Pediococcus eani, spelled O-E-N-O-C-O-C-C-U-S, O-E-N-I. It's a little ropey guy, and they're tiny, like, if I have a yeast cell, it's like a string, like this size, next to me. They're tiny, little microbes. But what Pediococcus do is they basically, they eat malic acid, they turn it into lactic acid. They eat malic, they pee lactic and fart carbonic acid. They make a little shit. And they, in the process of taking that malic acid, which is a fairly strong acid, they turn it into a fairly weak acid called lactic acid. Malic acid, for those who care, weighs in at about 134 grams per mole, whereas lactic weighs in at about 90 grams per mole. Not that that matters a whole lot. But malic acid is a much more stronger, it's a significantly stronger acid, meaning it's much better at pushing its proton into solution to get the pH down. Lactic acid's pretty weak, it's kind of wimp, it doesn't do a very good job of getting the pH down.
So this would be why the PV
Shifted
shift
food.
was so insane.
Absolutely, because you had 4 grams of malic. You had basically like over half, like 2/3 of the acid that's in the Riesling was malic. So super high malic numbers. That's why it's important to know those numbers, because if you have a wine come in and it's like you get a TA 7 grams later and you're all excited that you've got this great acidity, and then you look at it and it's all malic, you're a deep doo-doo, because you're gonna end up with a 3.5 gram a liter TA wine. It's gonna be like pH 5 and taste like the, yeah.
So with that conversion of molecular weight, does that correlate with the higher heat output?
No, it doesn't have much to do with heat, but what it does do is that difference in weight is the CO2 that's leaving the solution.
Okay.
That's what's floating off. And then attached to that CO2 is one of those electrons that is, it labels to lower pH is going out, and CO2 is carbonic acid, it's just leaving the wine. So that's why your pH rises, it's just leaving. You're letting acid flow it out into the air. And so the things that affect malolactic fermentation, there's 3 ways of approaching it in terms of how we do it. There's what we call co-inoculation, there's mid-inoculation, and end-of-ferment inoculation. So some people like to do, add their malolactic bacteria along with their yeast at the same time. And the science says this is the best way to do it. And if you're a person that's just into making healthy micro, this is the way to do it, for sure. Because number one, there's no alcohol around. Bacteria aren't gonna have to fight. They're just gonna be there. They're gonna be hanging out. They're gonna have a good old time. Second thing that happens is that yeast are making it warm. They like it when it's warm, so they're nice and comfy warm. Bacteria can't warm things up. They're not good at it. The yeast are, they warm things up. So it makes it warm and comfortable. It's like a little hot tub for bacteria. They like it. And then the next thing that happens is that yeast makes a lot of CO2. And the CO2 grabs these little tiny bacteria, this big, and throws them up in the wine. And they're getting thrown all around. They get tons of access to all the ml bacteria. That is bacteria. And why this is so awesome is that the population just grows massively. And yeast kind of limit, ML bacteria kind of don't. They just kind of rip. And you get a big population, and ML fermentation goes way fast. As a matter of fact, a lot of times it finishes right around the time you go primary fermentation. And why that's awesome is you go from crush in a red wine to finish in a barrel, buttoned down, in like 10 days. That's nice. Because then you're sulfured, you're safe, you're done. And, yeah.
Can you repeat the three? So co-inoculation,
Mid-ferment inoculation, and then end. Mid-ferment inoculation has basically the same advantages. But both mid-ferment and beginning ferment stand to have the same problem. The problem with yeah, I've got to make sure I'm doing this right. I believe, make sure I got this right. Sorry, my brain's a little short with a second. But Enococcus and I are what we call a homofermenter, which means they will go for not only malic, but they'll go for other stuff. And so what it means is that when they run out, their preferred food source is malic acid, but when they run out of malic, they'll move on to anything else. And so when they move on to anything else, they'll move on to, first thing, the next thing they go to is citric acid, and that makes the diacetyl aroma. They'll eat aldehyde, but they also eat sugar. And if the pH is above pH 3.6, that magic number again, they make acetic acid. And so if your yeast give up and you've got a rip-roaring malolactic fermentation, you can get a massive spike in volatile acidity. I am planning on doing DA so we can go over this again, so I'm just going to stay up short on this. And then that's your big risk. You're doing things in a short, that's your risk is that you can end up IPA with a stuck fermentation. The upside though, is that, now the other way you can do it is at the end of ferment. You get to the end of ferment and you inoculate with your MLS. And the reason you do this is because it's kind of traditional. You get the wine dry, then if you get a stuck ferment, you deal with that. Get that cleaned up, and then you add your ML bacteria and take it from out. There are heterofermenters too. So you know, heterofermentative ones, there's some heterofermentative lactic acid bacteria that just only eat. I think there's some lactic soils that really completely. And I always get them mixed up, so just leave me. So if I mixed it up, YouTube, I'm sorry. Rich DeCenzo can call me out later. The reason we do it after is that we've dealt with all the issues, and then we're going to have a very extended malolactic fermentation. It's going to take a long time to go through. It can take 2 months to finish, which means your wine's unsulfured for 2 months, which is kind of scary because there could be other stuff growing too. But remember how everything's like simmer and stew? If you've got some really nice new oak barrels, and you want to knit up all those oak aroma compounds, you want to allow your wine to hang out with a little extra acetaldehyde around for those tannins to get all knitted and polished up. You want those tannins to get knitted and polished up and integrate the oak and simmer it all together and make a nice round fleshy wine that's got all the right flavors. Then That's why you inoculate later. It's a risk-reward thing. Yes, you do have higher risk that that wine's going to be unsulfured for longer. Yes, it's going to take longer. And that's going to take more monitoring, more paying attention, more labor, more hours, more stuff. But your reward is that you'll end up with a more well-integrated and put together wine. That's essentially the nature of the beast. Now, for malolactic fermentation to work, we kind of focus on a few different things, and this is what TAPS is. So TAPS is temperature. Malolactic bacteria are very sensitive to temperature. They want to ferment it, they want to do the job at 70 degrees. They don't want much more or less than that. You get below 60, they don't want to go. You get below 50, they won't go. But also, conversely, if it gets hotter than 80, it gets hard on them, especially if there's alcohol around. So really, you're very temperature sensitive. So if you're in a cellar that has a hard time controlling temperature, it's really important to be able to keep your tanks at around 70 degrees. So you'll find a lot of wineries, even fairly nice ones that have a lot of aquarium fermenters, or aquarium heaters, that they put in their barrels. And they'll hang them in there and keep their barrel at 70 degrees to get the mallow complete. So that's the temperature. So that's one thing. So if you ever want to if you're trying to diagnose why your malolactic fermentation isn't working, temperature is one. Alcohol. They're a bacteria. They're sensitive to alcohol. You get up to 16% alcohol, good luck getting it through mallow. Good luck. Good news is if your wine's that ripe anyway, there's probably not much malic acid around. It's mostly gone anyhow. But you're going to sharpen. Malolactic bacteria are very happy at 0% alcohol, up to about 14, and very few strains land at much higher. Like it's really hard. So that's the stressors. P is pH. They're a bacteria. Bacteria is sensitive to pH. If you have a pH 2.9 wine, good luck getting it to go through malolactic. But in Champagne, they do take a lot of their wines through ML. Because there, it's not unusual to have a pH 2.9 wine. But they want to bring it up to a whopping 3.1 through malolactic fermentation. We don't have that problem. That is not an issue we have anywhere in the US. So we're trying to keep our pH in line. That's our biggest fight here, is to keep our pHs down. That's never going to be an issue for us. So very rarely will you see any sparkling houses in America be taking anything through ML. You take anything through ML? Ever, Gruet, or? No.
Not on purpose.
Yeah, I get that. That's also true. Good luck. It doesn't just happen. And that's one of the challenges with making sparkling wines, because sometimes you're making low alcohol and you're keeping your sulfur down, and sometimes it just comes. Whether you like it or not, it just shows up. I like to think that microbes are everywhere. And I have people that germaphobes make me laugh. I'm like, you can't get away. You can't live. You're a symbiotic organism with literally billions, if not trillions, of different bacteria inside of you that make everything work. So then the other factor is SO2. So bacteria are very sensitive to SO2, and so anything up to about 50 parts total is about as high as you can get and get a wine successfully through malolactic fermentation. Because what happens is SO2, remember we use that to bind aldehyde? So what happens is if a little bacteria, they're not very smart, they go along and they see this aldehyde because it's a sugar that, oh look, I can go eat something. They don't notice the Trojan horse on the backside of it, which is an SO2 molecule, and they pull it into their cell and it kills them. And then they die. It's very sad. I don't know, but it's like the Trojan horse. So SO2 content, we got to keep careful. So if you ever try to diagnose this ML, this is what we're talking about as TAPS. So temperature, pH, temperature, alcohol, pH, SO2. Then in that sweet spot in the middle, somewhere under 30, somewhere around 70 degrees, your pH above 3.3, and your sulfur, 10, just something around those range keeps you really happy to get it through. So that's a whole other fermentation. But I don't have ML on here because it was not in my attention, because in a lot of white wines, it's not necessarily something we're doing a whole lot. Outside of some Chardonnays and maybe some Viogniers, most whites don't. I would say the general whites don't go through a lot of malolactic. And the only reason we do it is it's traditional, and it makes the wine stable. Because once you got rid of all the sugar and you got rid of all the mallow, not much is there's not much there left to grow. So those wines are more stable for long term, and that's how we were able to ship wines as long as we needed and everything like that, the way we forgot that we used to do it, so. That's kind of it in a nutshell. Questions? Sorry, I was thinking, does that work? Too bad.
What's the plan tomorrow?
The plan domani, let me look at my, look at the to-do list. Okay, I'll do it right now. This, my brain, after I talk like this, I forget, I become a complete mess, so I kind of go into this weird zone where I don't. Yeah, it's good. Could you, Esther, could you just hit that off button on my phone? Tomorrow, I'm hoping we get all of our stuff.